Curaré is a leaf that grows on a vine in the canopies of the South American rainforest. The roots and stem are crushed into and added to other plants and poisonous animals. The paste that is created is dipped on the edges of arrowheads and darts. It is primarily used in hunting game. It has other functions. It's not just a toxin. Also used for edema, fever, and kidney stones. And if you do ever come across curare, do not fret. If it's ingested, it will not kill you. It must first get into the bloodstream. It begins with an action potential traveling down the axon of a motor neuron. Once the action potential reaches the synaptic terminal of the motor neuron, calcium ions enter the synaptic terminal through calcium channels. The rush of calcium into the terminal causes the synaptic vesicle to release acetylcholine, or ACH, into the synaptic cleft through the process of diffusion. From there, the ACH molecules bind to receptors, which are always there and on the muscle cell membrane. This action causes the cell membrane to allow sodium ions floating in the synaptical cleft to enter the muscle cell. This rush of sodium ions into the muscle cell through sodium channels causes the charge within the cell to depolarize, and another action potential is released. Inside the sarcomere of the muscle cell is an actin filament with trophomin and tropomyosin wound around it. Calcium ions that are stored inside the muscle are released as a result of the new action potential. These calcium ions then bond to TNT, altering their shape and exposing the actin filament. Myosin filaments that exist between the actin filaments and the sarcomere have energized myosin heads that attach to the exposed actin filaments. The myosin heads then pull the actin filaments, which causes contraction to take place. An ATP molecule then attaches to the myosin head, which allows it to release the actin filament. As the myosin head releases, the ATP molecule is broken down into ADP and P. The myosin head is now energized again, and the cycle continues. When curare molecules enter the synaptic cleft, they block the ACH receptors, which doesn't allow the ACH to bond to them. Because of this, sodium ions are unable to enter the muscle cell and depolarize it, which prohibits the action potential from being created and therefore not allowing muscle contraction to take place.